Photosynthesis is the process by which light energy is converted into chemical energy. This fuels ecosystems and replenishes the Earth's atmosphere with oxygen. The rate of photosynthesis, which is what we will be testing today, can be measured by either the disappearance of substance or the accumulation of product. This is the equation for photosynthesis. Necessary materials include sodium bicarbonate, which is just baking soda, liquid soap, plastic syringe, leaf material, a hole punch, plastic cups, a timer, and a light source. Our independent variable will be the bicarbonate concentration, and our dependent variable will be the number of discs floating after a minute. Control variables include the size of the leaf discs, or the amount of time a cup is under the light bulb, etc., etc. To begin, prepare two 300 milliliter bicarbonate solutions. One will be a control with 0%, so that would be no baking soda. The other will be a 0.2 solution, which is about 1 gram of baking soda in 100 milliliters of water. Cut 10 or more uniform leaf discs for each trial. Put a small amount of the sodium bicarbonate solution into the syringe and tap it around to suspend the leaf discs. Next, hold your finger over the syringe opening and draw back on the plunger to create a vacuum. Hold this for around 10 seconds and swirl the leaf discs at the same time. Then let off the vacuum. You may have to do this a few times, but the final goal is that the bicarbonate solution will infiltrate the air spaces in the leaf and cause the discs to sink. Now, pour the discs and the solution into a plastic cup and add the bicarbonate solution to a depth of about 3 centimeters. Now, place both the control and 2% solutions under a light source with a heatsink and start a timer. At the end of each minute, make sure to record your data by writing down how many floating discs you can see. You can continue to run this experiment with varying factors. For example, we chose to test a 3% and 5% solution. Our experiment concluded as such, with the green dots being the control 0% solution, the blue dots being the 2% solution, the red dots being the 3% solution, and the black dots being the 5% solution. We can therefore conclude that an increased bicarbonate solution will result in increased photosynthesis, albeit very slight. These results can be explained by photosynthesis, which uses sunlight, water, and carbon dioxide to create sugar and, as a byproduct, creates oxygen. Because the leaves have been deoxygenized, they will begin to float once they complete photosynthesis and oxygen is released, thus oxygenizing them. Once they begin to float, we can mark that as a point at which photosynthesis has been completed. In samples with more baking soda, and thus more carbon available, we can see that the reaction occurs at a slightly faster rate to that of photosynthesis elsewhere. Photosynthesis was slightly faster in samples with more baking soda, presumably because the abundance of carbon was detected by the plant cells, which realized they could now perform more photosynthesis with these materials. Because more photosynthesis could be performed, the plants likely chose to speed up the process to make best use of the higher carbon concentrations in the given time. Unique variance within the dataset can possibly be explained by differing distances from the light source, variation in original oxygen level/capacity, or possible unknown, unforeseeable contaminants within the water sources. Most of these are uncontrollable. However, the light source distance could be better controlled in future trials. In this case, the distance varied as the lamp had to be lifted to exchange the test beakers with other test samples. Outside of the laboratory, we can see this in leaves floating on the surface of bodies of water due to it containing some degree of the oxygen released by its photosynthesis. Using the experiment's data, one could know that living leaves would undoubtedly float eventually after they have been deoxygenized. Another application outside the laboratory would be that of a plant in a region with lots of water, carbon, and sunlight. These plants would photosynthesize faster than a plant in a region where these resources were less available. This is supported by the concept of photosynthesis being slightly faster with more carbon available, so it follows that more resources would mean faster photosynthesis in the wild.